This is the first of 2 nitrogen cycle videos. In this video, we're going to introduce the nitrogen cycle and talk about ways to make ammonia. And then in the next video, we will learn how to make the ammonia into amino acids. So the nitrogen cycle is super important because it is how we can get nitrogen gas into a usable form. Only bacteria can do this. And so without bacteria, we would just be in a world of disaster because we couldn't make amino acids. So the nitrogen cycle is super important. We're going to learn the 3 steps with stars. That's why they have stars. But there are 2 main parts that we are going to learn. So the first part is going to be how to make ammonia, as I said in this video. And then the second part is going to be how to use the ammonia to make amino acids. There are 2 basic ways to make ammonia. First is nitrogen fixation. So nitrogen fixation makes ammonia from nitrogen gas. Second is assimilatory nitrate reduction, which makes ammonia from nitrate, NO3-. The reason, well, we'll come back to this. Okay, so this is where we are headed. Okay, so first, nitrogen fixation, which again is the production of ammonia from nitrogen gas, which, if you don't remember, is 2 nitrogen atoms triple bonded together. Again, this can only be done by bacteria. This is really important to note, including cyanobacteria. And so some of the oxygenic photosynthesizers can do this. Bacteria that can do this are called diazotrophs. And so if you read somewhere, this bacterium a diazotroph, then you will know that that means that it can fix nitrogen. So nitrogen fixation is done by the nitrogenase enzyme complex. The nitrogenase enzyme complex is a complex of actually multiple enzymes. I think that there's 2. But people they always work together, and so people just call them the nitrogenase enzyme complex. One very important thing for you to know about this is that it does not work when there is oxygen. So very unfortunately, I've completely forgotten to print out the reduction potential chart. But I'm confident that you guys will remember that oxygen is at the bottom and nitrogen is not. And so if oxygen is present, then the electrons that are needed to disrupt this triple bond will instead be more likely to go to oxygen. And so the nitrogenase enzyme will not work in the presence of oxygen. And so oxygen must somehow be excluded from the local area around the nitrogenase enzyme in order for the enzyme to work. So this reaction will not occur in aerobic environments. So this is a written out equation for nitrogenase enzyme complex. What you'll need to know is that this takes nitrogen gas, and I've suddenly realized I haven't the slightest idea where these electrons come from. Some mysterious electrons and some ATP to give us ammonia. So you guys may remember that I've been talking a lot about how to fix nitrogen is really much more expensive than to fix carbon. And so although this is not technically an organic nitrogen, it still is a biologically available nitrogen. And so once you get ammonia, then a lot of different organisms can do assimilation. And so making ammonia turns nitrogen gas, which is not biologically available, into a biologically available form of nitrogen. So that's what's called nitrogen fixation. Nitrogen fixation with the nitrogenase enzyme complex requires 16 ATPs, which is 8 ATPs per nitrogen, in contrast to the Calvin cycle, which requires 18 ATPs but makes a 6-carbon glucose. So on a per atom basis, to fix nitrogen is way more expensive for the cell than to fix carbon. Sorry, I got distracted by this oxygen thing. So as a reminder, the nitrogenase enzyme will not work in the presence of oxygen. So, you guys, this is super exciting. A lot of the bacteria that do this live symbiotically in the root nodules of legumes. Legumes are like beans, peas, and peanuts. I always mention peanuts separately because a lot of people don't know that peanuts are secretly a bean. Also, a lot of people don't think of peas as a bean. Anyway, they're all the same. Beans, peas, and peanuts are legumes. And alfalfa. So legumes can fix, well, they can't, but legumes have in the roots these what are called root nodules, these clusters where bacteria live, and these bacteria can fix nitrogen, right? So these bacteria are called diazotrophs because they can fix nitrogen. And so in these nodules, the plant provides sugars to the bacteria, and then the bacteria will provide ammonia to the plant. But these nodules are red, and this is the awesomest thing ever. And so what is actually making these nodules red is something called leg hemoglobin. So exactly like hemoglobin in animals, leg hemoglobin will bind to oxygen. And so the job of leg hemoglobin is to exclude oxygen from the root nodules so that the nitrogenase enzyme complex can work. All of the organisms, the diazotrophs, all of the bacteria that fix nitrogen have different ways to get rid of the oxygen. But I think that this one is cool because it makes the nodules red. And I thought you guys might remember it more easily because it's like hemoglobin in the legume, so leg hemoglobin. Okay, moving on. This is just a list of various bacteria that can fix nitrogen. So rhizobium, we will come back to this for the final exam to learn more about specifically how these interact. But rhizobium are the bacteria that live in the root nodules of legumes. Anabina and nostoc are cyanobacteria. So if you will remember, hopefully as soon as I said cyanobacteria, you said to yourself, oxygenic photosynthesis. So anabina and nostoc both do produce oxygen. They also can fix nitrogen. And so they have specialized cells. You can see in this picture, this 1, 2 kind of yellowish cells. So these specialized cells are called heterocysts, and they do not produce oxygen, and oxygen is excluded from these cells, and that is where the nitrogen fixation happens. There are also some chemotrophs that can do nitrogen fixation. This is a different picture of root nodules with this nifty Y shape, so they can be kind of different shapes. Anyway, so these are organisms that can produce ammonia through nitrogen fixation, which again is done by, let me write it over here on the side, is done by nitrogenase enzyme complex, which turns nitrogen gas into ammonia and will not work if there is oxygen. So that's nitrogen fixation in a nutshell. Oh, okay, so I'm super excited to tell you guys about this. Let me see if I can zoom in some. So during the Holocaust, there is this guy named Fritz Haber. So Fritz Haber actually, I think, was Jewish, but worked with the Germans and was trying to basically come up with ways to kill lots of people. So he came up with like mustard gas and a bunch of really horrible things. But he also came up with the Haber process, which is a way to fix nitrogen gas into ammonia in a factory. And so Fritz Haber came up with this process. A later person with the last name of Bosch scaled up the process so that it could be reliably done on a large scale, giving us, I don't know if you guys can see this, the Haber-Bosch process. And so the Haber-Bosch process is super important because the Haber-Bosch process is how we get commercial fertilizer. And so if you have ever in your life eaten something that was not fertilized by nitrogen fixing bacteria or by animal poo, then you have utilized Haber-Bosch nitrogen. And so what I think is super cool about this process happened in the early 1900s, and so this dotted line is percent of the world's population fed by Haber-Bosch nitrogen, and you can see that as it goes up, the population growth rate goes up. This dashed line here is the estimated population if there was no synthetic nitrogen, and then this is meat production because, of course, meat animals like cows and chickens and whatnot eat lots of plants. And so in order to grow lots of plants, you have to fertilize them. And fertilizer, Haber-Bosch process. So super important. Also accidental. So Haber was like not excited about this at all because he was not trying to save the world by feeding them. But he wrote it down anyway because he was a chemist, and somebody else later figured out that it was super useful. So that was nitrogen fixation, making ammonia from nitrogen gas. The other option to make ammonia is called assimilatory nitrate reduction, and it is making ammonia from nitrate. So assimilatory nitrate reduction, your book makes this huge big deal about how this is assimilatory nitrate reduction and not, and not dissimilatory nitrate reduction. All right. So to assimilate something, right, is to bring it in. And so assimilatory nitrate reduction reduces nitrate with an end goal of making ammonia. Dissimilatory nitrate reduction also reduces nitrate, but with an end goal of making nitrogen gas. And so there are multiple different enzymes involved in both of these processes. It's not like you give electrons to nitrate and like you either make ammonia or nitrogen gas, but giving electrons to nitrate is the first step in both of those processes. The process that we care the most about, because it is the one that makes ammonia, is the assimilatory nitrate reduction process. And this has two steps. Let me see if I can zoom in over here. Step one is nitrate, which is NO3 minus to nitrite, which is NO2 minus, and this is done by an enzyme called nitrate reductase. You can see that the electrons come from NADPH, and then they go to FAD, and then they go in. The second step is actually a two-part process, and we're just going to say that it goes from nitrate to ammonia, or from nitrite, sorry, to ammonia, and it is catalyzed by nitrite reductase. This can happen, as you can see here, in a variety of different organisms, super importantly, not in animals. So, assimilatory nitrate reduction, the other way to get ammonia initially from nitrate.